Hello and welcome to part 2 of the pentose phosphate pathway in gluconeogenesis. And in this video we're going to focus on gluconeogenesis. So the learning objectives of today's video is to understand how the body uses non-carbohydrate precursors to make new glucose. This process is called gluconeogenesis. So what is it? Is the metabolic process by which organisms will produce sugars, namely here glucose, for catabolic reactions from non-carbohydrate precursors. It's important to understand that gluconeogenesis is not simply the reversal of glycolysis. On paper they may look the same, but they are two distinct biochemical pathways. Glycolysis is the conversion of glucose to pyruvate. This process generates ATP, whereas gluconeogenesis is the conversion of pyruvate to glucose, but this process requires 4 molecules of ATP and 2 molecules of GTP. So you can see that gluconeogenesis requires more energy than is generated by glycolysis. And some of these reactions are not energetically favorable, and this is why gluconeogenesis requires the extra energy. So if you remember from the glycolysis lectures, we had some key catalytic points, and these were phosphofructokinase, hexokinase and pyruvate kinase. And these are distinct enzymes which are not involved in gluconeogenesis. So what we have in gluconeogenesis, we have counterparts which are distinct gene products, and these will catalyze the reverse reaction. So for phosphofructokinase, its counterpart in gluconeogenesis is called fructose 1,6-bisphosphatase. For hexokinase, we have glucose 6-phosphatase, and pyruvate kinase has 2 enzymes that are required for the conversion at that point, and they're called pyruvate carboxylase and phosphoenolpyruvate carboxykinase. So what does the gluconeogenesis pathway look like? Again, we start from bottom to top. We have pyruvate, which goes through more or less the same molecules as glycolysis, eventually ending up with production of glucose. And the key regulation point, the first one, is glucose 6-phosphatase, and remember that in glycolysis on the right hand side, this reverse reaction here is catalyzed by hexokinase. So what we're doing here is we're removing the phosphate from glucose 6-phosphate to produce glucose. We have fructose 1,6-bisphosphatase. Again, on the right hand side, the counterpart would have been phosphofructokinase, and this removes a phosphate group from fructose 1,6-bisphosphate to produce fructose 6-phosphate. And at the bottom, where we have a single enzyme which converts phosphoenolpyruvate to pyruvate in glycolysis, and this is catalyzed by pyruvate kinase. We have 2 enzymes that perform this function, phosphoenolpyruvate carboxykinase and pyruvate carboxylase. So what can we make glucose from? Well, the non-carbohydrate precursors will include lactate. Remember that lactate is produced by glycolysis in the absence of oxygen. So lactate can be converted back to glucose. We can also use amino acids, and these will come from your diet or from the breakdown of proteins when you're not getting sufficient glucose from your diet, and also from fatty acids such as glycerol, and we get this by the hydrolysis of triacyl glycerides. Each of these non-carbohydrate precursors will enter the gluconeogenesis pathway at different points. So lactate can be converted back to pyruvate through the action of lactate dehydrogenase. The amino acid aspartate can be converted to oxaloacetate through a transamination reaction. Alanine, again, will enter at a slightly different point, and alanine can be converted to pyruvate. And the fatty acids, such as glycerol, can enter about halfway up the chain. So glycerol will enter at the point of dihydroxyphosphate acetone, and this is catalyzed by a phosphorylation oxidation reaction. So it's important, because of the energy requirements of glycolysis and gluconeogenesis, that both pathways are not active at the same time. So if glycolysis is active, gluconeogenesis is inactive, and what this does is avoid wasting energy, because we don't want to be producing energy and using energy at the same time. If these reactions were occurring at the same time in the same cell, what we would have is that glycolysis would be generating 2 molecules of ATP, whereas gluconeogenesis would be using it. And so what we'd have is a net energy loss of 2 ATP and 2 GTP molecules. It's important to note that most of the gluconeogenesis, the metabolic pathway, takes place in the liver. So this is where your body will generate new glucose. Small amount also occurs in the kidneys. And so the liver is important for maintaining blood glucose levels so the brain and the muscles can function properly. So often lactate from active muscles will be pumped into the bloodstream. It will go around the body into the liver, and the lactate will then be converted back to glucose, and the glucose can then enter the bloodstream, which will then feed the brain and your muscles. So we're going to look at the key control point, which is fructose bisphosphate, when it's converted to fructose 6-phosphate. Again, remember that this enzyme is fructose 1,6 bisphosphatase. This enzyme catalyzes the hydrolysis or removal of the phosphate group. And similar to phosphofructokinase, the key modulators are the same. So we have ATP and citrate, which will stimulate the activity of this enzyme, whereas AMP will inhibit it. And fructose 2,6 bisphosphate, remember this comes from the conversion of 1,6-phosphate, will also inhibit this enzyme. So whatever activates phosphofructokinase will inhibit fructose 1,6 bisphosphatase, and whatever activates the bisphosphatase will inhibit the phosphofructokinase. This ensures that glycolysis and neogenesis don't occur at the same time. So here is a table that I've constructed just for you to view in a visual form of exactly what is happening. So ATP will switch off ATP will inhibit phosphofructokinase, so switching off the enzyme, whereas ATP will activate the 1,6-phosphatase. Conversely, AMP activates phosphofructokinase but inhibits the 1,6-phosphatase. Citrate will inhibit phosphofructokinase but activate fructose 1,6 bisphosphatase. And the molecule fructose 1,6 bisphosphate, remember, which is generated from the 6-phosphate, will activate phosphofructokinase but inhibits fructose 1,6-phosphatase. So why do we need to generate new glucose? Well, as we've just mentioned previously, is that it's important for maintaining your blood glucose levels so that both your brain and your muscles can function properly. It's also important to note that the glucose that your body requires is not provided by your diet. Not all of it is so we use other ways of getting energy from non-carbohydrate precursors. But to do this, we need to convert it to glucose. And it also provides a way of the body of recycling so-called waste products of glucose metabolism, such as lactate. Active skeletal muscle will produce lactate and alanine. These can then be released into the bloodstream and converted in your liver. The lactate and the alanine are then converted by your liver back into glucose. So lactate is formed from pyruvate during anaerobic respiration. And the conversion of lactate back to glucose is what's called the Cori cycle. So this takes place in your liver. So here on the right we have an active muscle which is converting glucose to pyruvate, which in anaerobic conditions is converted to lactate. This can then enter the bloodstream. It's then taken up by the liver and then converted back to pyruvate and then eventually to glucose, which can then be released into your bloodstream to maintain your glucose levels. In this way, the metabolic burden is shifted from the muscle to the liver. So the gluconeogenesis is not taking place. Remember, the gluconeogenesis is very energy demanding, so it requires a lot of energy, and it shifts that burden from your muscles, which is requiring the ATP for motion, so to move your muscles, and it puts the burden onto the liver. Active muscle also converts pyruvate to alanine by a transamination reaction, and this is called the Cahill cycle or alanine cycle, and this occurs when your muscles are being degraded. What happens here is that here we have the Cori cycle, but when we get protein degradation, we get production of alanine. Alanine is then taken up by your liver. Alanine can then be converted to pyruvate, and pyruvate then to glucose. So when your muscles are atrophying and you can get breakdown, so it's you basically using proteins as a source of glucose. So hormones play an important control of gluconeogenesis, as they do for glycolysis. So insulin is released, and this will promote glycogen synthesis and glucose uptake. So glucose uptake is in your liver, and it will then produce fresh stores of glycogen. Insulin will also inhibit gluconeogenesis. Remember, insulin is released when you are well fed, and so there's plenty of glucose around. So you don't need to produce new glucose. So insulin will lead to the inhibition of gluconeogenesis. As well as inhibiting the pathway itself, and it does this, insulin inhibits gluconeogenesis by reducing the protein level of two key enzymes, the phosphoenol pyruvate kinase and glucose 6-phosphate. So it reduces the amount of enzymes that are actually present. Glucagon obviously will have the opposite effect, so this will inhibit glycogen synthesis and promote gluconeogenesis, and it does this by raising cyclic AMP levels in the cell, activate protein kinase A. This will activate an enzyme called phosphofructokinase 2. This phosphorylation of this enzyme converts the enzyme from a kinase to a phosphatase. The phosphatase action of this enzyme then cleaves fructose 2,6-bisphosphate to fructose 6-phosphate. Remember that fructose 2,6-phosphate is a key activator of glycolysis. It stimulates the activity of phosphofructokinase. So any reduction in the 2,6-phosphate reduces the activity of the phosphofructokinase and therefore promotes the activity of fructose 1,6-bisphosphatase. This enzyme, of course, is involved in gluconeogenesis and thereby promotes the metabolic pathway of gluconeogenesis. Multiple choice question. So which one of the following statements is correct in gluconeogenesis? We have A, glucose and NADPH are produced. B, insulin stimulates this pathway. C, lactate, alanine and glycerol are converted to glucose. D, lactate amino acids and acetyl-CoA are converted to glucose. Or E, pyruvate amino acids and acetyl-CoA are converted to glucose. I'll just give you a moment to pause the video if you'd like to have a go at the answer. Okay, and the answer is, of course, C. So in gluconeogenesis, no NADPH is produced. Remember, NADPH is involved in the pentose phosphate pathway. Insulin inhibits this, it doesn't promote this pathway. Lactate, alanine and glycerol are all converted to glucose. Remember, the lactate, alanine and glycerol are the non-carbohydrate precursors which are required to produce new glucose. Next question, so which one of the following is correct in gluconeogenesis in the liver? And here we're looking at hormonal control. So A, glucagon inhibits the pathway and AMP stimulates. B, glucagon stimulates the pathway and AMP inhibits. C, glucagon stimulates the pathway and ATP inhibits. D, insulin stimulates this pathway and AMP inhibits. Or E, insulin stimulates the pathway and ATP inhibits. I'll just give you a moment to pause it if you'd like to have a go at the answer. And the answer is B, glucagon stimulates this pathway and AMP inhibits. So why do we look at these metabolic pathways in detail? Well, future clinical directions for looking at this sort of pathways is looking at, say, type 2 diabetes, which of course we all know is a metabolic disorder. It's characterized by an insulin deficiency, insulin resistance, and excess endogenous glucose production. So if we can switch off or at least reduce the amount of gluconeogenesis, this is going to be beneficial for the patients. So fructose 2,6-phosphate is a key enzyme in the generation. Remember, the counterpart in glycolysis is phosphofructokinase. So inhibitors of this enzyme are being researched at the moment as a potential treatment for type 2 diabetes. There are many clinical trials underway and new drugs are being developed all the time. And it also begs the question of what other additional targets exist in the glucose metabolism pathways that could be used to treat this type of disease. And if you'd like to read any more about gluconeogenesis, then I suggest chapter 18 of Straya. And thank you very much for listening.